Hello, my name is Caterina Gratton, and I'm an associate professor in psychology and neuroscience at Florida State University. In today's presentation, I'll be overviewing some of our projects on the use of individualized approaches to study networks associated with the frontal lobe. Many folks have long been interested in the frontal lobe, especially the prefrontal cortex, given its connection to high-level functions, including working memory, attention, and inhibition. These functions are disrupted trans-diagnostically across a range of psychiatric and neurological disorders, including schizophrenia, depression, ADHD, and Parkinson's disease. While many have focused on specific regions of the frontal lobe and how they contribute to these processes, seminal work from Patricia Goldman-Reich and Marcel Mezelam, as well as others, have established that specific frontal regions are interconnected with one another and other portions of the temporal and parietal cortices, forming distributed networks that have a parallel and stereotyped topology and contribute to complex functions like attention. We can study these networks non-invasively in humans using functional connectivity methods that many of you will be very familiar with. These track statistical dependencies in the bold signal between different regions of the brain, for example, the rows and the columns of this matrix below. And networks defined with these methods can recapitulate many of the observations from previous track tracing work and make clear that a large number of closely interspersed networks are present in the frontal lobe. These patterns have also been shown to be quite repeatable across studies, despite differences in data collection and analysis methods, including in the observation of control networks like the frontoparietal, shown here in the yellow and orange colors, and the cinguloopercular, shown here in purple and lilac colors, respectively. These methods have also made it clear that networks associated with the frontal lobe are not only connected with regions in the frontal lobe, but also regions of the parietal and temporal cortex, as well as stereotyped locations in the cerebellar, thalamic, and striatal locations. However, these findings I've reviewed so far have largely been built off of studies that assume group correspondence. That is, that the same coordinate location will represent the same functional location across individuals. This is problematic as, again and again, locations of the frontal lobe have been identified as among those with the highest variability across individuals, across a number of different data sets and age regions. To illustrate the import of this observation, let me give an example from a recent review written by my former postdoc, Derek Smith. In it, he discusses the case of the medial frontal cortex and dorsal anterior cingulate, which is perhaps one of the most argued about regions of the brain. In a neurosynth analysis, if you were to query different terms, you can see that many different cognitive functions are associated with these regions. And we can also see in a zoom in on the group network average that there are many different networks present. However, the situation is further complicated when one looks at the network structure of individual people, where we can see that networks are varying in their exact locations and extent across individuals. This means that many past studies using group averages or group-defined regions are likely mixing networks across individuals, that is, joining information about apples and oranges in their results, which may contribute to the wide variety of different results associated with, or different functions associated with these regions. One approach to addressing these issues is to shift towards more individualized models of analysis. For example, rather than collecting a relatively modest amount of data on each participant, precision fMRI methods are based on collecting extended amounts of data on a smaller sample of individuals. This large amount of data can then allow researchers to produce individual network maps, like I showed you on the previous slide, and show that these maps are very reliable with test-retest samples. We can then use these sort of techniques to identify and characterize locations of individual differences, much like I showed you in the earlier slide. This supports the observation that individual differences are particularly prominent in the frontal cortex, especially in the right hemisphere. Interestingly, these individual differences come in different flavors. While many are associated with shifts in the borders between networks, some are also associated with isolated islands of individual differences, as investigated by my research tech, Ali Doretsky, who has shown that it's particularly common to have these isolated islands in caudal frontal regions. Using these methods, we also asked whether individual differences are important for how the brain coordinates its functions across different brain regions during task states. Particularly, our graduate student Alexis Porter took a machine learning approach based on individual data in the Midnight Scan Club. She trained a machine learning classifier based on 9 out of the 10 days from Midnight Scan Club participants to classify among 5 different task states. She then tested this classifier on independent left out data from that participant, the 10th session, for example, or on independent data from another participant. And she can use this data to then ask, first off, is it possible to create individualized classifiers to predict task state? And to what extent do they generalize? And what does that tell us about task states in the brain? So Alexis was able to show that individualized classifiers are indeed successful. They can predict task state both within as well as across other participants, suggesting there's some degree of generalizable performance. However, as you may notice by looking at this graph, classifiers perform far better within the same participant than they did in other individuals, suggesting that there are person-specific components to task state. And indeed, when we look at the features used by each of these individualized classifiers, we see that they differ quite a bit across different people, but not across different cross-validation runs within a person, suggesting that there are person-specific features to task states that can be revealed with these individualized methods. We can also use the information from individualized network maps to guide improvements in group level analyses. For example, my research tech, Ali Duretsky, analyzed a large set of individualized network maps from several high data datasets and used these to create a probabilistic map of brain networks across people. So here, for example, each color represents the percentage of people that have the default mode location in a particular vertex. And Ali was able to show that we can identify both regions of high consensus as well as a penumbra of regions that have more variability across individuals and many common networks of the brain. She then used these maps to create a series of high probabilities ROIs that you can use to seed future group analyses and have high confidence that these regions are part of the same network across individuals in your study. And also to create a point-and-click tool that can be used to probe the probability that any given location of the brain is associated not only with, say, the first network that's most likely in that location, but the second and tertiary network and so forth. And this can be used to interrogate past results to understand the individual variability in the common networks that are present at a given location. So hopefully this will be useful to some of you. I put the GitHub link here on the webpage. Do please feel free to use it and reach out if you have any questions. I'll just provide one example of how we used this in recent work to probe control networks of the brain associated with the frontal lobe, and in particular, the cinguloopercular control network, where we use some of these high probability locations to probe the extent to which control network functions are consistent across different locations once we've controlled for individual variability across people. And what we find is that there are indeed regions that show very consistent control functions, for example, to cues during tasks, error-related activity, sustained activity during tasks. But interestingly, this is only true of a subset of regions in the cinguloopercular network. And the cinguloopercular network actually seems to divide up into at least 2 distinct subsets, one of which is associated more with control functions, including in a neurosynth analysis, and the other of which is associated more with motor and interoceptive functions. So hopefully this presentation has suggested to you that it's important to study networks in the frontal lobe, but that it's particularly useful to take an individualized perspective to these questions. I've shown you, for example, that individual differences are particularly strong in the frontal lobes, especially these differences that are isolated and not well addressed by close spatial transformations like border shifts. One approach to addressing these individual differences is to use extended sampling precision fMRI techniques. And just as an example, I showed you that these techniques can reveal individual differences across brain states. I also showed some examples where we can use these precision fMRI techniques to then inform and constrain group studies by the use of probabilistic maps, where we've seen that, for example, finer scale specialization can be found within the cinguloopercular control network. I'll just close by thanking my collaborators on these projects. I've highlighted and read the folks whose work I highlighted during the talk today. Please reach out. I have my contact information here if you have any questions, or we're also recruiting. So if you're interested in joining our group or have suggestions for others that might be interested in joining our group. Thank you very much.